So the life cycle of a sample, once the customer puts the samples in the mail, it's going to be received at our facility, and then it's going to go through essentially an inventory.
We're going to log that sample into our system so we know that we've received it, and then we're also going to connect it with the animal information that you've sent. So it's very important for us to understand, we got this tissue sample, who is the animal inside of that tissue sample, right? The sample is only as valuable as understanding the animal inside of it. When we make that connection, that kind of starts the processing within the lab.
As soon as we know that we have all the information, it should come into our lab that same day. Then one of the members of our team will go ahead and handle that individual sample and put it into a plate.
We're just getting that tissue sample, that hair card, that blood card, a section of it into the plate.
Samples are only touched the one time. They're plated in the location and they live their entire life cycle throughout the lab in that one location. They'll go through a digestion period, an extraction period, and then they'll go through the Illumina process, which is what we use to drive creating our genotypes. When we look at our process, we've limited the amount of times that a sample moves. We're reducing the risk all the way across the process to ensure that the sample that we got in is the genotype that we put back out.
Those processes will end up denaturing the DNA. It takes those two strands and unravels it, and then we add the A, C, G, and T, those 4 building blocks of the DNA. We take those and use our primers, and we add that to the denatured DNA to help build out those structures.
And what we're trying to visualize is actually the underlying genome that sits on that animal. Through that custom array, we've picked pieces and parts that we find are most valuable and important that actually turns that DNA result into those traits and those indexes that you can use to actually make a decision on an animal.
Our turnaround time is really targeted around that 10-day mark, right? And so we understand that it's a priority from customers that these decisions are back in their hands fairly quickly, but there's also some of the balancing that we have to do because of the science behind it.
There are times in the process where science can limit us. For example, sit in an oven for 16 hours because that's how long it takes for the science to happen. There are pieces and parts within the process that we can actually adjust, because we've got people that are responsible for it and they can shrink that turnaround time.